All right, so this lecture is going to be about the basal ganglia, starting with the anatomy of it. So when I say basal ganglia, if we're talking about, if this is like a cross-section of the brain, the basal ganglia are all located right here, sort of right in the center. They're very deep, and it's where a lot of the cell bodies themselves are, sending out axons in different areas. So that's why they're called the deep gray nuclei. And so what are the different pieces? Well, I think there's really 6 main pieces that you should know at the medical student level. So the caudate, the putamen, the globus pallidus, which is, there's 2 pieces to it, the external and the internal part, the thalamus, and just below that the subthalamic nucleus, and the substantia nigra. And we're going to go through each of these pieces, try to visualize where they are and how they work together. So I really think that it helps if you start with a 3D model. I think too many times when you're being taught neuroanatomy, they just show you cross sections, and it's really difficult to understand how they all kind of piece together. So I have a 3D model here of all the main pieces that we need to really understand and see together. So if I rotate it a little bit, you can kind of see how all of these are arranged in space. And I think that's going to make it a lot easier to understand why the cross sections look the way they do. And I think if you can keep this picture in mind, it'll all become very clear. So let's rotate this back a little bit, and let's start by orienting ourselves to things that we know. So actually in this picture, we actually have here, these 3 are the brainstem. So let's start with that. So right here in red, the midbrain. Underneath that is the pons, and then the medulla. So now if we're actually going to go talking about the main pieces of the basal ganglia, we're going to have to go a little bit higher up here. So these were all the nuclei that are involved. Let's start with these gray structures here. So you see there's one here on this side and there's one here, and you see how they loop around and kind of come around like a C shape, right? These are the lateral ventricles. They're filled with CSF, right? And actually, right next to the lateral ventricles, you can see this structure here looping around. That is actually the caudate. And right next to the caudate, this big oval structure here, that is the putamen. So we can see our brainstem, lateral ventricles. Right next to the ventricles is the caudate, and then a little bit of space, and then there's the putamen. And I want to rotate just a little bit more to the side, and I'll keep the labels on for some of these. So again, you can see the lateral ventricles we have here in gray. Right next to it is the caudate, and right next to it is the putamen. So next, you'll see underneath the putamen, there's these little structures. So there's one there, and there's one there and there. And actually, these are the globus pallidus, globus pallidus. And you can see that there's an external and internal piece on both sides. And they're basically touching the putamen. So keep that in mind as well when you see a cross section. And then here and here in green, we have the thalamus, which sits right underneath the lateral ventricles, directly underneath it. And you can see how all of these are sort of arranged together. Now, I mentioned 2 other pieces in the beginning of the lecture, the subthalamic nucleus and the substantia nigra. So let's just rotate a little bit more, get a front-on view. So again, this is the thalamus in green, and the subthalamic nucleus is going to be just below the thalamus. So it's not in this 3D picture, but if I were to draw them in here and here, just below the thalamus would be the subthalamic nucleus. And then the last piece that you should see is the substantia nigra. And this is actually, as you can see, it's in the midbrain. So that's where these are all located in space, related to one another. So now, if we try to look at a cross section, a lot of it makes a lot more sense, right? So let's start with just sort of orienting ourselves. So very often you're given this. This is a coronal slice. And so if we draw in here, you can see here and here, these are the lateral ventricles. And you can see if we go to the 3D picture, how that can kind of look just like that. If we were to slice it down the middle, this is the type of picture that we would see. And then right next to the lateral ventricles, remember we said is the, right here, the caudate. And you can see how it's right next to it here, right? And then there's a little bit of space right here, and then the putamen. And again, a little bit of space here, and then putamen. And then right underneath the putamen, we have the globus pallidus, external and internal. And again here, external and internal. And here in green, we have the thalami. And remember, it's not drawn in this picture, but I'll just draw it here quickly. These would be the subthalamic nuclei. You can see them here, just below the thalamus. And then, in the midbrain would be the substantia nigra, which you can see here. So now I think a coronal section makes a lot more sense. There's only one more thing I wanted to discuss, which is this space here and here. So this space is actually called the internal capsule. And what is the internal capsule? Basically, it's just a bunch of axons. So this is actually just like a whole bunch of axons coming down from where? From all the different pieces in the cortex. And all of those axons from the cell bodies of the cortex, they all come down and they go through right here. And that's how they descend down into the spinal cord. So if we were to look over here at a 3D model, they're coming down like that. Just to kind of make that piece seem a little bit more clear. And if we want to see that in 3D, because you want to see how sort of, why does it make this slanted shape like that? Well, if we see it in 3D, again, we have the cortex here, and all of the cell bodies are sending down their axons. And here we have the thalamus right in the center, right? Our lateral ventricles would be right there. Globus pallidus, external and internal, and then the putamen right outside of it. So if you remember, in between the caudate and the putamen and the thalamus is where this would all run. So you can see how all of these fibers come and they all kind of run right in between and they go down. And this is coming out towards you, right? This is coming out in the Z plane. So you can see how they would create, when you look at a cross section, sort of this arrow shape. All right. And lastly, I just want to show you from the side. So I've taken this diagram here. This is a axial cut. So if you look over here in our diagram, we're cutting the brain like that. And I just kind of rotated this picture. And again, I have the 3D model on the right. So I want you to try to understand why it would look like this if you were to cut the brain right there, right through the middle. So you can see how we have our lateral ventricles here, wrapping around. So if you were to cut across, right across the center of the brain, you would have just a piece of the lateral ventricle showing. So this part is actually here, right? But there's also the back part of the lateral ventricle, which you can see here and here. And then, right next to the lateral ventricle is the caudate. And then a little bit of space where our internal capsule would run through. It's right there. So this is the internal capsule. And then you have the putamen right here. And then underneath the putamen, you can see it sticking out a little bit here, is the globus pallidus. And you can see how if you went just a little bit farther in, you would be at the thalamus. And that's it. So hopefully that orients you a little bit towards what the pieces are, where they're located, and sort of how to remember them arranged in 3D space so that when you see a coronal or an axial cut or any kind of cut, you can kind of orient yourself to say, okay, this is where it is and this makes sense. This is how it would look if it was like looping around in 3D space. So next time we're going to talk about the actual functions of each of these pieces and how they connect to each other in our physiology lecture.